And we're not done yet, right? We can also use ultrasound imaging to see the structure of hair follicles, such as the Arrector pili muscle, which the paper titled, quote, Ultrasound Image of Skin Apparatus and Imaging Basics, unquote, by Molsek and Malinowaska in the Journal of Ultrasound Sonography, shows us, right? You can see a picture of the Arrector pili muscle. We can also note the sebaceous glands and other structures that are noted in another study by Wortsman et al. Titled, quote, Ultrasound Characteristics of the Hair Follicles and Tracks Sebaceous Glands, Montgomery glands, apocrine glands, and Arrector pili muscles, unquote. Here we can see a side-by-side with an ultrasound image and a biopsy of the tissue. The utility of ultrasound here is breathtakingly apparent, I would say. And it's kind of cool that we can get a sort of image in ultrasound like this of the Arrector pili muscle, because it is theorized that in androgenetic alopecia, or in the end stage of androgenetic alopecia, where hair loss theoretically is not reversible, if the Arrector pili muscle disconnects from the hair follicle, then there is a sort of permanent hair loss. And this is mentioned in the paper titled, quote, Beyond Goosebumps, Does the Arrector pili Muscle Have a Role in Hair Loss, unquote, by Rodney Sinclair and colleagues, where this crucial function of the Arrector pili muscle is explored. And in order to prove that, maybe we have some sort of intervention that is seeming to, activate hair follicles from dormancy. Let's say PP405, for example, right? Let's say it actually does that. Can we note that there's some sort of structural change to the Arrector pili muscle? Is the Arrector pili muscle reattaching itself? Maybe, let's say, if verteporfin actually does regenerate hair follicles, is it recreating a new Arrector pili muscle? Like, what's going on here, right? So an ultrasound imaging technique could kind of help us elucidate that without having to do any sort of biopsy. I think all of this is important because we may not have to be invasive in such a process. And if the process is more refined, we can get a better understanding of hair loss disorders on a large area across the scalp, again, without having to do multiple biopsies. We could also combine other methods to analyze scalp disorders. So let's use Lichen planopilaris as an example. This is a scarring or cicatricial alopecia, and cicatricial just means scarring or fibrosing. And here in Lichen planopilaris, the immune system attacks the sebaceous glands due to a defect in a receptor called the PPAR gamma receptor, which causes for an excessive amount of lipids to build up in the tissue and create lipotoxicity. This causes for the lymphocytes and other white blood cells to come in and attack and destroy these tissues, releasing cytotoxic substances in this process, which may be indicative of an autoimmune attack. You can get an understanding of this process by reading these 3 research papers titled, quote, Lichen planopilaris and frontal fibrosing alopecia as model epithelial stem cell diseases, unquote, by Matthew J. Harris et al. The second paper would be, quote, Lichen planus, unquote, by Katharina Bach et al. And finally, the third paper here would be, quote, PPAR gamma agonist and their role in primary cicatricial alopecia, unquote, by Harchuong and Shachuanat, where you can get an understanding of the PPAR gamma receptor and how its downregulation can cause Lichen planopilaris, but how certain drugs like pioglitazone, if it agonizes the PPAR gamma receptor, it could potentially treat Lichen planopilaris and restore function to the hair follicle. So instead of doing a biopsy to identify Lichen planopilaris, we can get an understanding of the scalp by getting a large scalp ultrasound mapping of the scalp to see the areas of inflammation, as well as Doppler coloring of that ultrasound to see where excessive blood flow pooling is, which is indicative of, again, inflammation. And we can look at another study titled, quote, The Suitability of High-Frequency Ultrasonography, 20 megahertz, in Evaluation of Various Forms of Primary Cicatricial Alopecia in Relation to Trioscopy, Pilot Study, unquote, by McKeel et al. And in this paper, researchers used 20 megahertz in order to evaluate the scalp of healthy individuals, as well as diseased individuals with hair loss disorders. They noticed that high-frequency ultrasonography could effectively differentiate between the structural changes associated with various types of primary cicatricial or scarring alopecia, against healthy scalp conditions. The study meticulously analyzed the echogenetic and morphology of dermal and subdermal layers, assessing elements such as entrance echo, follicular structures, and dermal-subdermal borders. So essentially how the echo interacts with different parts of the tissue and how different borders are defined to say, hey, this is a hair follicle and this is an inflamed area. Just a really, really good study, and it has some pretty nice images here too. Individuals with active forms of cicatricial alopecias displayed specific patterns like cigar-shaped hair follicles, as seen in Lichen planopilaris, and frontal fibrosing alopecia, as well as discoid lupus erythemia, and this is a pretty bad hair loss condition. And to kind of emphasize how good this imaging is, right? These patterns were very distinct when compared to the ultrasound imaging of healthy people that didn't have any sort of hair loss issue or follicular damaging. So it gives us some really good highlights on, yes, ultrasound imaging, especially at high frequency, is very potent in helping us understand the skin structures without having to do any sort of biopsy. So we can get real-time recording without having to excise the area, so that's a really good thing because it's really not invasive at all compared to the biopsy.